Welcome to your lesson on photosynthesis and the basic structure of plants. Now, we've already watched this video, the amazing photosynthesis video. It's a good visual for you to be able to see the bigger processes of what's happening. It's not got a lot of details, though, so we're going to run through more details that are grade 12 university level worthy. And we'll start off with talking about chlorophyll itself. Photosynthesis is a special process broken into two sections. You have the photo stage, which we'll talk about first, obviously, because it's the first stage. And then we'll talk about the synthesis stage. The photo stage is where light is involved, and the synthesis stage is where carbon dioxide becomes important. Because we're actually going to be grabbing that carbon dioxide and we're going to be fixing it or attaching it to other molecules in order to create the C6H12O6 glucose sugar that we're going to need. So we're taking the carbons from the carbon dioxide and attaching them. The type of organisms that are able to perform photosynthesis have to have the some version of this chlorophyll molecule. So plants have it, some versions of algae has it. You have protists, which are, if you remember back from grade 11, there was the 6 kingdoms. So you've got the animals, and you can pretty much always identify what an animal is for the most part. You've got plants, easily identifiable. Fungus, okay, good. You've got bacteria and archaebacteria as 2 separate kingdoms. So eubacteria, archaebacteria, great. But then there's this dumping ground kingdom called protista. And it's basically the idea is if it's not quite an animal, it doesn't quite meet the definition, they call it a protist. If it's not quite a plant, but it's sort of like a plant, but it's not enough to be a plant, it's a protist. And the same thing with fungus. So within the protista world, you have a huge variety. And it's my guess that probably in the next couple decades or so, they'll probably split protista as well. That's what I think they would do. In high school, when I was in high school, we were still using the kingdom Monera. So your parents as well would have known different kingdoms. And the Monera would have covered probably the protista world, and the bacteria kind of would have been thrown in that kind of general area. Monera was kind of just all the little things. So even in the last couple decades, things have changed a fair bit. Okay. So point being, you have to have this special pigment literally paint, so pigment, called chlorophyll. And what it's going to do is help us to absorb light energy. There are 2 major types of chlorophyll that we will look at. Chlorophyll A is kind of a bluey-green spectrum. That's the type of light it's going to absorb. You'll see spectrums down below. Chlorophyll B is in the yellow-green range for what it absorbs. A is known as the primary pigment. So tuck that fact away because that'll come up again. A is the primary light-absorbing pigment. And if we want to get away with not writing the whole word, we say that the short form is chloro-A. So what do they have in common and what is different about the 2? Well, they all have this porphyrin ring. That's what this big structure is here. You do not need to know the structure of this. All you need to know is that there's chlorophyll A, chlorophyll B. I'm showing you the structure so you understand how it gets embedded into the phospholipid bilayer of the chloroplast. Okay. So it has this head here called the porphyrin ring. And this little R group sitting right here makes all the difference which functional group you put, because if you have this little CH3 group, then it's chlorophyll A. But if you have this group, the COH, carbonyl group, it's going to be a chlorophyll B. And just having that slightly different part in that one little section determines which wavelengths of light it can actually absorb. So then this is why functional groups are so important. Everything else about the molecule is identical, but that tiny little change changes everything. Again, that's why we started off our first unit talking about functional groups and why they're important. So now you're going to see it here. In the center is a little tiny piece of magnesium. It's an ion, so it's been stripped of its electrons. And it's surrounded by this hydrocarbon ring. And then you've got this long chain here, this hydrocarbon chain, called that because it's made of carbons and hydrogen, so it's a hydrocarbon chain. Do you remember with the fatty acid chains, we said that if you have a phospholipid, let's say, you could have a charged head, but you have a non-polar hydrophobic tail sticking off of those phospholipids. So you have the little head with the 2 legs. Those 2 tails have the similar idea where it's a bunch of carbons and hydrogens. So this would also be non-polar and hydrophobic. And that's great for us because it means that when I compose this molecule and I build it and I put it into the cell, this part here is very happy to tuck itself in and hide inside a membrane, and this part here is very happy to stick out the top. So the structure of it allows it to stay stuck to the membrane. inside, like anchored in there. It's quite brilliant, actually. Without the tail, the chlorophyll wouldn't anchor. Okay. We have other types of porphyrin rings in nature. Lots of them, actually. This is just one version of it. If you were hemoglobin, you might have this porphyrin ring, minus the tail, but the porphyrin ring with a piece of iron inside it instead of magnesium. So other cytochromes you're going to see as well when we talk about the mitochondria and how it works with its membranes. So cytochromes are just big rings of stuff that are going to help us to pass electrons. They'll absorb energy and they'll throw it away as an electron and pass it to the next thing. Okay, we'll get to that when we get to that. The differences then, again, as we said, is just that little methyl group. But the main difference that we care about, I don't need you to memorize the methyl and the aldehyde. The main difference that we care about is what part of the light spectrum are they able to absorb. So let's take a look at that graph now. So what you're looking at here is the light absorbed. How much of that light will be removed. Remember in grade 10, you learned that the way that light interacts with things is it can strike an object, and then the object, depending on its characteristics, can either absorb the energy, and it can convert it to heat or whatever. So a black object absorbs a lot of light and convert it to heat. That's why your car will heat up a heck of a lot in the summer, whereas a white object would reflect all that. So you have absorption and you have reflection. The other thing that could possibly happen is that it could be transparent and the light could pass through. But in this case here, we're talking about the absorption side of it. What wavelengths of light do you absorb? I want to remind you as well from grade 10. Let's say we're looking at somebody wearing a green sweater. Okay. If you can see green, what wavelengths of light have been reflected back to your eyes? Green. But remember that you have red, green, and blue available. So what is not reflecting back? If you can see green, red and blue must be absorbed by that material. Makes sense, right? Hopefully. So what we're seeing here then is that within the purpley bluish section, we're seeing a lot of absorption from all three of these different types of pigments, and just a little bit of absorption down here towards the red end, but no absorption in this particular area, especially from chlorophyll A. Look how there's nothing being absorbed here. That's why chlorophyll appears green, because look at what's not being absorbed. If it's not being absorbed, it is being reflected back. Make sense? Then what we see reflected back from chlorophyll B is just a slightly different color. We have a little less blue in the light, but still it's more of a greenish part of the spectrum bouncing back. So both chlorophylls are going to appear green. And then we get this third type of particle called a carotenoid. Carotenoids are called accessory pigments. It's another color that's in there. When you see the word carrot, I want you to think oranges and reds. That's actually, carotenoid is the pigment that's in carrots. It's what makes them look that color because they're very high carotenoid content. And if you look at its spectrum, it's from cutting off from the green that way, it is reflecting back all of this yellowy, orangey red light, and that's why it appears within that spectrum, within the yellows and the oranges and the reds. Okay? But it's absorbing in this particular area. Notice that chlorophyll A actually only absorbs a very narrow area, a narrow band of wavelengths. Chlorophyll B does its own separate band of wavelengths that it's really good at, and the carotenoids have a very wide band. Here's why. The chlorophyll A is going to only be able to absorb specific parts of light, and the chlorophyll A, being the primary special pigment, is the only one out of these three types that's actually able to give up an electron so that the electron can begin to bounce through a membrane. And you'll see what the electron ends up doing. But the whole point is we want to give up an electron here. Chlorophyll B doesn't give up an electron, but what it will do is it'll take the energy that it's been given by the sun and it'll convert it and pass it off to chlorophyll A so that chlorophyll A can get rid of its electron. And then the carotenoids will do the same thing. They're going to absorb a different set of wavelength of light, again, bounce it through it's called resonance transfer, we'll get there, and you give it to chlorophyll A so that chlorophyll A can give up its electron and you can start the process again. So chlorophyll A is my main one. Everybody else is just there to help. Try and get a wider range of energy absorbed so we can make the magic happen. Just a little bit of language so that going forward you know what we're talking about. Again, in grade 11, you should have learned the terms prokaryotic and eukaryotic. Prokaryotic is a very fancy way of saying no nucleus. So karyo is referring to a nucleus. So these guys don't have a nucleus. Prokaryotes are your bacteria. And specifically, when we're talking about the type of bacteria that's able to harness the energy from the sun, we're talking about cyanobacteria. If you remember as well from grade 10, a lot of access from grade 10 here, you would have learned that cyan is that bluey green color. So cyanobacteria actually appear that bluey green color. All right, that's why they're called cyano. An autotroph is something that is self-energy maker. That's what that means. Auto means self. So on their own, they can make energy. They don't have a nucleus. Move forward in genetics unit, we'll talk more about these guys. So there's a huge group of them based on their numbers, not based on the different kinds of them, but they're probably the most abundant thing on Earth. And these are what are forming your algae blooms. Anywhere that you have too many nitrates or phosphates, which are basically fertilizers, when those get thrown into a pond, you'll have these guys will bloom out of control. Have you ever heard of the red tide? Let me show you a picture of red tide. Okay, so here's an example of red tide. These algae have been given so many nitrates and phosphates and stuff that their actual population has just grown out of control. With these particular ones, if you were to go into the water during a red tide, you would actually end up with like diseased skin. It would damage skin and cause rashes and things like that. But what it does to lakes, even if it's not a red tide, even if it's another type of algae bloom, we call it algae bloom, what it ends up doing is it sucks up so much of the oxygen from the lake that the fish actually begin to die and all sorts of bad things begin to happen. So they end up using up too many resources, and the plants, and they're quite thick sometimes as well, they form a thick mat, so that the plants underneath don't have the right access to sunlight, so then they're not producing enough oxygen, and then things begin to die. So algae blooms, no good. You don't want an algae bloom. You cannot swim in an algae bloom. If you see anything like this, don't go near it. If you've ever seen pig farms as well, I don't know what that is all about, but okay. That's pretty cute, though. So this will happen as well in farmers' fields when you see something like this, where there'll be a runoff from the field itself, and it'll go into these ponds that are then, there's pond all in a row, and it's kind of like filtering out all the garbage from things. This is one way to protect the actual streams and rivers that are near a farm from dumping all of this stuff straight into the water system itself. So the water kind of drains into this pond, and then the good water will seep out of the pond into the next pond, get purified again by soil, kind of like a filtering system. But this is what some of the ponds will look like if you've got a lot of fertilizer being used on your fields, and specifically pig poop does this. Okay, in general, and you got a lot of nitrates and whatever. Okay, let's go back to our note. Okay, so prokaryotes, basically what we're saying is that they are also able to have this chlorophyll molecule, and they're able to make their own food. The eukaryotic autotrophs that we're talking about then would be the cells that do have a nucleus. So EU means true or real. So you have a true nucleus, they are making their own energy, and these would be your other categories. So you've got your plants, you've got the protists that are plant-like, so they're photosynthetic, and then the algae. And these are the ones that will generally look more green because that's the wavelength of light, again, that is being released or reflected off of that. You want to commit this to memory, and you'll also want to commit the reverse of it to memory as well, because the reverse of it would be cellular respiration. Okay? So in general, what we're going to do is take some carbon dioxide. This will actually not happen until the second stage. We're also going to take some water, and we're actually going to rip water apart. Water is made up right now of hydrogens and an oxygen. And what we're going to do is blow it apart, not just into hydrogens and oxygens, but we're actually going to take the hydrogen itself and rip that apart as well. And we do that with a special enzyme that's built into the chloroplast membrane. Well, the thylakoid membrane inside the chloroplast, but we'll get there. So we're going to split this into 3 parts. The water with the help of light energy and the enzyme that does the job called the Z protein or Z protein, depending on what your favorite way is to say that letter. So you're going to end up with half of an oxygen, half of molecular oxygen, so 1 O in other words. You're going to end up with a couple protons and a couple of electrons, and this would be the hydrogen atom blown in 2. Because remember, the hydrogen atom is 1 proton, 1 electron, no neutrons for the most part. The vast majority don't have any neutrons. So if I split it in half, I'm going to have a proton, and we always represent that with the H+, and then I'll have a couple electrons. These electrons are going to be workhorses for me. They're going to be pass through a membrane and do a whole bunch of jobs of ending up in another molecule to make it high energy. They're going to go off to another stage to help us fix carbon dioxide. And then the protons are going to be used to drive a pump. and that pump is going to make another energy molecule for us. So it's going to be very useful. The oxygen needs to get out of this particular thing. You don't want it in the chloroplast or particularly in this cell building up, because it actually slows down the process of photosynthesis. But I want you to remember that all organisms go through some version of cellular respiration. So not only is the plant doing photosynthesis in this direction, but the sugar that it makes and the oxygen that it produces, it will also reverse that so that it can get a different version of energy out of it as well. It's creating sugar for its own benefit and oxygen for its own benefit, but it makes so much of it. It's such an amazing producer that literally everything else in all of the green earth is actually able to benefit from the fact that plants are amazing at capturing sunlight and turning it into sugar. So remember this formula here, commit it to memory. And also please remember it as a balanced formula, so that if you're going to make 6 carbons over here in a glucose, you better put in a total of 6 carbons on this side. So make sure you memorize the balanced one. It's easy because it's 6 and a 6 and a 6. And it matches your glucose molecule. Okay? So light energy, I've put it onto the arrow because light energy is not actually a reactant. Reactants technically are all the atoms that you throw into the pile, right? And so you should always get whatever atoms you put in, you should get atoms out. The law of conservation says that we don't create and we don't destroy the matter. We can convert things, but we don't destroy it. So every carbon that went in has to come out and so on. Light energy doesn't actually contribute any matter because it's energy. So the special condition is that you have to have light energy available, so it goes on the arrow, and you have to have this chlorophyll available, so it goes on the arrow as well as a special condition. If you end up putting plus light energy, though, I'm not taking the mark off. I'm just telling you this is the more better way to write it, because that's good English. Okay. The other thing is you're going to see as we move forward, more so in the other details, but this oxygen that is produced at the end here is not from the carbon dioxide. I know we talk about that you exchange gases, carbon dioxide for oxygen, and that does happen in your lungs, but to actually produce this oxygen, it has nothing to do with carbon dioxide and everything to do with the water. Every single one of these oxygens came when we split the water. And then all of these carbons then, plus that oxygen there, is coming from my carbon dioxide. Okay, so just keep that in mind as we move forward. That's a lot more detail than you really need to know for this particular lesson, but it'll help you in the long run. So let's jump now into the basic structure of it. So when you're looking at a cross-section of a leaf, and this again was nicely shown in the video that we saw, you can see we've got this upper layer called the cuticle. Cuticle is made of wax. It's basically, we call the wax on the edge of your fingernails cuticle as well, and that's just a protective layer. Wax is a type of fat, and so it's waterproofing. So not only does it prevent water from coming in, it prevents water from escaping, which is the main thing, so that the plants don't dry out really, really quickly. It allows them to suck water up when they want to, and it also gives them control over how much water is being transported up, because all the water that they lose, at least hopefully, if they're in control, the water that they lose is going to fall out of the holes at the bottom here. The holes are called stoma for one, or stomata for many. So you've got the hole at the bottom. On either side of the hole, there's something called a guard cell, the 2 guard cells there. And we're able to cause, well, we aren't. They're able to cause those guard cells to either swell, which actually opens the hole, makes more of a bean shape, and it forms a hole in between, or to become flaccid. There's that word. So either turgid and they're open, or flaccid and they become a little bit limp, and that'll slam them shut. And that way it's able to open and breathe, or close it and hold its breath. The reason it might want to hold its breath is it might be losing, not only exchanging gases through that hole, but it could be losing too much water through that hole as well. Water vapor can fall out. So if the plant is feeling like it's starting to dry up, it can send a message to the guard cells to say, we need to close up just for a little bit. It's too hot. I'm losing too much water. And they can control that. So it's basically they hold their breath, and then they're able to open again when the conditions are right. We'll talk about that in detail at the end of our photosynthesis unit. So you got your cuticle on both sides. Then you got the upper epidermis and the lower epidermis. That's a fancy way of saying the leaf skin. Basically, you have dermis as well. You've got 5 layers of dermis. They have 1 layer. Then immediately under that, and by the way, this upper epidermal layer and the bottom, they are so thin, they're actually transparent. So we're not blocking any light. because we want the light to go through these layers in order to reach this palisade layer. The reason it's called palisade is because of the way it's constructed. It looks like a bunch of columns. If you think about famous Greek temples or even just courthouses these days where they have the big pillars at the front, so you go up the steps, you have these big pillars and this open kind of like an area where people stand around outside the building before they go into the big doors. That's called a palisade with all of those columns. That's why it's called palisade here. Then you've got a layer under that where it has a whole lot of holes in it, so it makes sense then that we're going to call that spongy because there's holes in it. The palisade mesophyll layer is where you'll have the vast majority of your actual photosynthetic cells that are capturing light and doing the conversion to sugar in those particular cells. Then you also have a bunch of tubes that we'll talk about. Actually, we'll talk about them right now. We have two major types of tubes. You've got the xylem and the phloem. The reason I put numbers here, and you can copy these down, is that these are trying to point out the important differences between xylem and phloem. So xylem is a dead type of tissue. It's basically like an empty straw made of cardboard because cellulose, cell walls of these plants are still there, but the cells themselves are no more. So you basically got these cardboard tubes on the inside. You can call it a vein as well if you don't want to call it a vascular bundle, but its job is to specifically transport water and anything floating in the water like minerals that have been dissolved into that water. And it goes from the roots, where it has access to groundwater, up to the leaves. It is a one-direction process. So I'm not pumping the water. In fact, I'm transporting this water for free. And I'll talk about that when we hit transpiration down here. So it's a dead tube, transports water, one direction from the roots up to the leaves. The phloem, on the other hand, is alive. So it's actually able to pump things in one direction, and then if it changes its mind, it can pump things in another direction. It's specifically important for carrying carbs. So any type of sugar that I produced, we will be producing glucose, but you don't only produce glucose. We're just going to simplify the grade 12 conversation and pretend like we always do. There are other sugars that are produced, but glucose is the main one. So any type of carb that I've created, the sucrose that I can put together to then transport it as sucrose, I can pump that back and forth using the phloem. And so it's from wherever I've done photosynthesis, usually up in the leaves, to wherever cell in the plant needs it. And if I have too much sugar produced, then I can store the rest of it. I was going to say for a rainy day, but a rainy day might be good for a plant, for a day when maybe they're not making as much sugar. So they can have lots of storage, or for us to come along and enjoy. And usually that is in the roots, although it depends on what type of plant you have. Things like potatoes create tubers. The actual potato itself is a tuber, and it's a specialized kind of a root that you have this massive storage thing. A carrot is mostly root because it has this massive storage area. Other ones will have little skinny roots, though. They may store some sugar there, but maybe the majority of their sugar is stored in their stem. It just depends on the type of plant. But to simplify it, we're just going to think about plants in general. And so we'll say it's from the leaves all the way down to the bottom is where you'll supply your sugar, and it can move in any direction that it is needed. Okay. So let's take a look again. Dead versus living is your first answer to what's the difference. Carries water and minerals or carries carbs is your second. And then the direction. One direction from the roots up to the leaves versus from the leaves to wherever I want it and then store it in the roots. In any direction I want to go, up or down. All right, so how does the xylem, being dead, actually transport anything? How do you get that to happen? Well, we have this process called transpiration, and it's a passive process, which means we don't need energy. That's very important. And what it really means is, if you were to ask what's transpiration, you would say it's a loss of water vapor from plants. It's when plants are losing water. You would say that you perspire when you sweat and you lose water, and we say that a plant transpires when it loses water. Why do we want to do that, and why would we ever want it to fall out those holes on the underside? Because water has this wonderful ability to stick to itself. There's two things you learn about water in grade 9, is that it is cohesive, sticks to itself, cohesive, and adhesive, sticks to other things. And so water being cohesive, again, because of the polar nature of it, where you've got the slightly positive hydrogens and the slightly negative oxygens. If you were to pull one little molecule of water, it's got this attraction to the molecule behind it. So you're kind of like you tug on one and you tug on them all. They kind of form this sort of tugging chain that you can do. So if I was to go back up to that picture of the leaf, if I have a water particle fall out the bottom here, it's not just randomly nothing else in there. We've been sucking up water through these tubes and delivering it to the space. So if water vapor falls out, it encourages another bit of water to move over, and then it ends up sucking all the way up the tube. So every time I use water or lose water, I just pull another one out from the ground. Okay, so our xylem then is going to use this transpiration process to help us to pull water up, and it's all for free. All I have to do is open a hole at the top, and if water falls out, all of this water gets transported. And if you're thinking about a tree that's 60 feet high, it is moving over 200 liters of water a day. 200 liters, that's 100 big pop bottles of water a day completely for free. If you had to pull 100 pop bottles worth, 200 liters of water up to 60 feet, it would take you a heck of a lot of energy to get it there. And this is free. Brilliant system, just using the basic nature of water to draw this up. And not only do I get water, which I need for photosynthesis, but I also get anything that's floating in the water transported as well. So I have the benefit of all of these minerals that I might need, so the nitrates and the phosphates, all this stuff that's going to help me to build stuff and to run chemical reactions, that all comes for free as well. Brilliant. It doesn't cost them any energy. The other thing it will do, not only will transpiration allow all this transport of water up to the top and any minerals that are in it, again for free, no energy expended, you will also at the same time allow cooling of the leaves to happen. I think you can imagine that if you were standing outside all day, exposed to the sun, you would probably want to sweat too. And so this is going to cause some evaporation to happen of that water. And what water will do, again, another wonderful thing about water is that it has a high heat capacity, which is a fancy way of saying it takes a lot to heat it up to make it turn into a gas. So if you can, it takes 100 degrees actually to go all the way from where it first becomes a liquid to where it becomes a gas, right? 100 degrees is a lot of energy. So if I can throw some of this heat energy at those water particles, and they can expand into that water gas, they can take that heat with them and I can cool down my plant. It's the exact same process that your body does when you sweat. You evaporate the water off your skin to take away some of that heat. Now again, we mentioned these guard cells. And guard cells are the ones that are controlling how fast the plant can exchange its gases, so bringing carbon dioxide in and losing oxygen out, and transpiration, which is losing that water vapor. Let me show you the picture of it. It's nicer if you can look at it at the same time. So here's the two guard cells, and this would be the hole in the underside of the leaf. And the basic idea is when I swell the cells, because they are this weird little bean kind of a shape, if you swell them, it actually opens the hole. The hole is called a stoma. If I allow them to get a little bit more flaccid, so I lose some of the water, they're not swollen, they're flaccid, then they flop closed. You can see it in the picture, right? You don't need to know all of these things in detail up here, but what I do want you to know is that we have full control as a plant cell over whether I'm opening or closing these. I can do it at will, according to the needs of my entire structure. So there'll be some kind of information system that will come and tell the guard cells when to worry about it. And in particular, just in general, usually it's activated by light energy that turns on a switch. So in the morning, you can see down here, in the morning, you'll have a bluish light that turns on a receptor in the guard cells. So they have little receptors built into their membrane that they can detect light. And then that starts something called a proton pump. Proton pump is really just a protein that's built into the membrane that's able to throw protons out of the cell. So if I start throwing away stuff, then what I can do is create something called an electrochemical gradient. We'll get more into this. It's basically like an active transport pump. You throw a proton on one side of the membrane, and that opens up the channel for another thing to come in. So throw one thing away that turns on the pump to bring something in. And in this case, what I'm doing is bringing in potassium ions, allow them to flow in. And when that happens, as soon as the potassium ions flow in, water follows. So what I'm really doing is making the inside of the cell hypertonic with this potassium, and then the water will just naturally flow into it. What if I want to go the other way? I throw the potassium away. If I can get the potassium out, the water will follow and it will shrink. So that's it. I'm controlling it, actively transporting in one sense and then allowing facilitated diffusion in another sense with the potassium ions. So I'm controlling which direction I'm throwing ions, water follows. That's it. So one more time. Throw ions inside, water will come in, they swell, I open the hole. Throw ions away, water will follow, it'll become flaccid, and you'll close it up. That's it. So there are more details in here that I need you to know. That's the basics of what I need you to know. Okay, water, just make it hypertonic, you'll swell it. Make it hypotonic, and you will lose the water. And if you're more interested in that stuff, you can read that through for yourself as well. Let's get into the actual structure of the chloroplasts themselves. They are on the micro scale, so they are measured in micrometers. Want to remind you, though, that micrometers are still pretty small. Micrometers are 1/1,000,000th of a meter. So take a millimeter, cut that into 1,000 pieces, and now you have micrometers. So they're still pretty small, but we can see them on the microscope. They're not as small as DNA or as a cell membrane and whatnot. So they're, as far as structures go on the microscale, they're pretty large. So they're about 3 to 8 micrometers long, about 2 to 3 wide, and they look something like this here. What's neat about them is that they do actually have their own DNA, which means they store information for some of the proteins that they're going to need, and they have their own ribosomes, so they have their own protein-making factories. There are some things that it's going to need the nuclear stuff for, but in general, it's not always relying on the nuclear information, that nuclear DNA, to run all of its processes. It is somewhat autonomous. You're going to find that with the mitochondria as well. Mitochondria has its own set of genes and can make its own proteins. Okay. They can also replicate on their own by something called fission. Fission is what bacteria do. Bacteria will grow. It's called binary fission. They'll grow, and then they just cut in two after they've replicated their DNA so that each half will have it. So it's very similar to bacterial growth. Fission just means to cut. So they replicate by cutting in half, and now I've got two. It's made of 3 membranes. You have the outer membrane, which is called the outer membrane. Then you have an inner membrane called the inner membrane. That's pretty easy. The main membrane that we care about, though, is something called the thylakoid. The thylakoid are these little coin discs that you kind of see here. And these discs are going to be where you have all the chlorophyll attached. At the beginning, I showed you the structure of chlorophyll. I showed you it had this hydrocarbon tail for burying itself into a membrane so that it could anchor itself. And it's the thylakoid that it anchors itself into. And there will be all this chlorophyll attached to the outside of the thylakoid with that little tail dug into the phospholipid membrane. Okay. So we would call this a membrane-bound organelle. Membrane-bound organelles are only in eukaryotes. So you wouldn't necessarily find this guy in, the bacteria would have to have its own version of this because they don't have membrane-bound organelles. I told you about endoplasmic reticulum, and it's called reticulum because it folds on itself. So we have membrane reticulations or folds in our inner space as well, and that's what the thylakoids are. They are folds and discs to increase our surface area. It would basically be like if you were to put your hand up and then stick your fingers together and then measure the surface area, you'd have so much surface area. But as soon as you open your fingers, now your hand has more surface area, but the same volume. You take up the same space, but there's more places. Now imagine that, same thing with like shelves on a wall. You have a wall and you have a certain amount of space, but as soon as you put 10 shelves on the wall, now I've got more space for stuff. Right? So that's what folds do. They increase our working space. And that's what this has done here, increased working space here. So let's zoom in now on the thylakoid itself. You're going to have the thylakoid. The one single one is called the thylakoid. The whole stack is called a granum. See this word here? G-R-A-N-U-M. U-M is one stack. All the stacks, or at least more than one stack, is called grana. So thylakoid is the disc. The grana or granum are the stacks of discs. And then you can see that they're joined by this, I don't even know what that looks like. It looks like a parasite. It looks awful. But this little thing here that joins them is called the lamellae or lamellae, or however you want to say it, lamellae. If it's A-E, that's the plural. If it's just A, that's the singular. Again, we're dealing with Greek and Latin endings of words to do plurality, so that's why we're getting those weird kind of endings. So lamella or lamellae, and the lamellae are basically very similar to a thylakoid. It's just that it joins the stacks. Same kind of an idea. Within a typical chloroplast, you have about 60 of these. So obviously this is not even close to showing what it would really look like. We don't have 60 here. We have, 9? And then in each of these stacks, you should have 30 to 50 of them. So again, that's not even close to showing you how many there would be. So there is a ton of these photosynthetic factories built in here, okay? So again, you've got the outer and inner membrane. You've got the thylakoid, which is a third type of membrane where all the photosynthetic magic is going to happen. If it's in a stack, you call it a grana or granum for one, and they are joined by the lamellae. That is your structure of a chloroplast. There's also juicy bits. Inside the thylakoid disc, you're going to see there's liquid inside of that, and that's called the lumen. So inside the thylakoid is the lumen. Lumen is just a word in general that means the inside. So we could talk about the inside of your intestinal tube as being the lumen of the intestines. It just means the inside. Okay? So lumen is a general term, inside. And then stroma is all the liquidy stuff that's kind of outside of the stacks, but inside the inner membrane. So everything else out here, okay? We're going to have two important processes. We're going to have lots happening in the lumen in the first stage, and then the second stage where we're actually using the carbon dioxide to make sugar, that'll all happen out in the stroma. You're going to need to know your locations. Again, photosynthesis is broken down into the photo stage, where we actually care about light and we use water. We need to have chlorophyll, light and water, oxygen gets created, and then we're going to produce or use these particular energy molecules. We're going to adapt them. We're going to add a hydrogen here with an electron, and we're going to add a phosphate here. We're going to use the energy of these final products, which are these guys right here, to drive the next set of reactions where we want to take carbon dioxide, shove it onto this rubip molecule, and eventually end up with something called G3P. P-gal is the same name. This is a half sugar. Half sugar is as far as we are going in the photosynthesis discussion in grade 12, but that's where we're going to end up. Let's take another look here. So now we're zooming in with our diagrams. So we have our leaf cross-section. We've got our epidermal layer with the cuticle, palisade layer, spongy mesophyll. You've got your veins with your xylem and phloem. You've got your underside, and then you've got your stomata with the, so stoma being the hole with the guard cells on either side, opening and closing that, so I can exchange gases. When I zoom in then on the actual chloroplast, so I've zoomed into a mesophyll cell, whether it's the palisade or the spongy, doesn't matter. So you zoom in on a chloroplast. This is a real picture, by the way. And then you see you've got your outer and inner layer, you've got your thylakoid, you have your grana, you have the lumen on the inside, and then you have the stroma on the outside. And again, this is another real picture, electron micrograph of what they actually look like. And then, if you actually looked at one thylakoid disc, fortunately we have to do it sideways, this is what is built into the thylakoid membrane. We've only shown one example of it, but you have these proteins and these things called cytochromes that are built in, and you got the chlorophyll attached here to this part of this particular photosystem. And you have electrons that are going to bounce through the membrane. They're going to end up in these molecules and get taken off to the stroma to do some work. And I'm going to have this pump over here pumping protons out to allow me to make some ATP. And we're going to focus in on that on the light-dependent lesson, which will be a little bit later.